Hey everyone, we're pivoting away from metabolism finally and getting into all things cell division and nucleic acids and whatnot. So we're going to talk about the cell cycle, talk about tumor suppressor genes and oncogenes. We're going to talk about epigenetics, DNA replication, the whole mechanism, an emphasis on telomeres and how they're related to cancer and aging. We're going to talk about DNA damage, what's a point mutation versus frame shift mutation, DNA damage repair mechanisms. We have NER, BER, MMR, double strand break repair, transcription and transcriptional regulation with an emphasis on the lac operon. And finally, we'll close off with RNA interference and CRISPR. So for the cell cycle, we're just going to go through the different phases of the cell cycle with an emphasis on mitosis. What are cyclins and how do they relate to CDKs? And what for the G1 to S transition, the checkpoint, there's a lot of important proteins, retinoblastoma, P53, E2F that we should know about. And then we'll just sort of characterize tumor suppressors, oncogenes, and try to provide like a nice framework to a lens through which we can see and think about them. All right, here's our cell cycle. So G1, cells growing, cells functioning, nothing to write home about. S, we are replicating our DNA. G2, cell is growing, cell is functioning. And then mitosis, the cell is dividing, and mitosis is divided into prophase, metaphase, anaphase, telophase, and then cytokinesis, where we are cutting it in half. Sometimes people have an extra prophase, prometaphase, but I don't think that's so crazy important. Some cells will go into something called G0. So you might go into G0 if there's just not enough nutrients around, and it's just, maybe it's a bad time to, the same thing with humans, right? If a human female is going through a point of starvation, amenorrhea, she might stop ovulating, and it's just because it just doesn't make sense evolutionarily to get pregnant. If you're starving, you can't support a baby. Similar with division. If there's not enough nutrients, it's probably not a good time to divide. So it's going to hold off. But other cells are just permanently in this differentiated sort of quiescent state, something like a nerve cell or a muscle cell. Additionally, so some things don't really divide ever, and other cells might divide very quickly. A skin cell, a GI cell, any sort of cell that, an epithelial cell, something that's interacting with the environment, where there's lots of, mutagenic chemicals, reactive oxygen species, a lot of acid in our stomach. These cells are undergoing a lot of damage, so we have to replace them. So they or their progenitors might divide a lot. And you can cells are constantly picking up mutations and accumulating damage, but division puts an extra stress on the cell, especially during S phase, the copying mechanism might introduce a lot of errors there. So the more division you do, the more likely a cancer is to develop. So we might expect skin cancers and GI cancers and whatnot to be more common than a cancer that affects nerve cells, right? We know about brain cancer, lots of different cancers of the nervous system. They're actually usually going to be the accessory glial proteins, like in a oligodendrocyte or a Schwann cell. The actual neurons, because they're in G0, not dividing often, don't often get cancer. Of course, they can, but it's not as common. So the checkpoints, when we're looking at G1, we mentioned, want to make sure there's nutrients, want to make sure there's growth factors. The rest of the body and the rest of the cells are saying, yeah, we think it's a good time for you to go ahead and divide. And there's just no DNA damage, not a lot. If there is DNA damage, let's try to repair it. And if we can't repair it, maybe we should commit apoptosis and take ourselves out of the equation. After G2, we want to just make sure that, S phase went according to plan and that there were not a ton of mistakes. And then after M phase, let's make sure that all of the chromosomes separated, correctly. We don't have any sister chromatids still stuck together, things like that. Everything that's not mitosis is known as interphase. So mitosis. So let's talk about prophase and prometaphase. So our objective is to, we got 2n equals 46. So we've got 46 chromosomes, and we want to put, we want to split the sister chromatid. So half go into one cell and half go to the other cell. The most effective way to do that, just imagine DNA is like a ball of yarn like this. It's really difficult to separate the genetic information if it's in this yarn form. So you can imagine that one thing's going to happen is we're going to want to condense the chromosomes. It's really easy to separate, or easier to separate the DNA if it's condensed. Instead of yarn all over the place, we've condensed it into a little like spool of yarn. That makes it easier. Number two is we need something to pull the DNA, from to one side of the cell and then the sister chromatid to the other side of the cell. So we have these little centrosomes, which just have, little just centrioles, I would say. these are going to be our fishermen, which are casting, the lines to grab onto the chromosomes and pull them to the other side. So it would make sense that the chromosomes are condensing so that we can separate them easily. Two, the fisher people are appearing and they're starting to cast their lines. And then three, well, the DNA is stuck inside of the nucleus, and how the heck are you going to separate the genetic information if it's stuck inside of the nucleus? So we're going to go ahead and see that the nuclear envelope is dissolving. So we see that in both of these. Metaphase is just all of the chromosomes are nicely lined along the metaphase plate. We have our centrosomes, our fishermen on opposite sides, and they have successfully landed their mitotic spindle, which are microtubules, on the kinetochore, on the centromere of this of these chromosomes. Anaphase is just we're pulling apart, we're reeling the fission, and then telophase is really just the opposite of everything we saw in prophase and prometaphase. The chromosomes can start to uncondense. We want the nuclear envelope to reform, and the fishermen have done their job, so they can sort of go away. And we see all of that happening in this picture here. Additionally, though, we have cytokinesis, where a cleavage fur will develop, and we're going to have microfilaments sort of like choke off the cell so that it becomes, if you were going to like cut some, let's say you're making bread and you want to cut the dough with like a sharp wire. That's effectively what's happening here, and so we're going to split the cell in two. So pushing the cell cycle forward, we have these things called cyclins, and cyclins cycle. So we have D, E, A, B in that order from G1 all the way to M. And these are going to correspond with the cyclin-dependent kinases 4 and 6 and 2 and 1. So let's just look at G1. It's going to start to be transcribed and translated, and we see that the levels are going to go up, 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 and they peak right towards the end of G1. Interesting. They're peaking towards the end of G1, and that's because they're combining with CDK4. CDK4 is cyclin-dependent kinase 4. So when it's paired with its partner, they can now try and push the cell cycle forward. We then see similarly E accumulating. E is going to accumulate with 6. And together, the complex is going to try to push the cell cycle forward. And then once they've done their job and they've transitioned us into S phase, they can start to be degraded. Now it's job for cyclin A to start accumulating, and it peaks right at the end of S. And you know what? That's because it's going to team up with CDK2, and it's going to push us into G2. And then we're going to see the same thing with B, pairing up with CDK1 and pushing us into the M phase. And now a specific example in something we're actually going to go into more detail on is we might see like cyclin D in CDK4, and something that we're seeing happening as G1 progresses is we have this retinoblastoma protein. And notice that it's getting progressively more and more and more phosphorylated. And once it is fully phosphorylated, then we are allowed to proceed from G1 into S. So the CDKs of G1, it's important that they phosphorylate RB for us to get transition into S phase. But you know what? If there's damage, we might not want to go through with division. One of our checkpoints isn't met. And that's where we're going to have something like the P16 protein or other P proteins inhibiting the CDKs, because they're, on the one hand you can think of them as wet blankets, but on the other hand they're we need them, right? They're saying, hey, like maybe it's a bad idea to divide if not everything's perfectly going according to plan. So this is actually an entire mechanistic scheme that we should be familiar with. So this I think is very high yield to understand inside and out. So E2F right here wants to push the cell cycle forward, right? It is pro-division. Retinoblastoma here is a tumor suppressor. It is inhibiting E2F. That's its natural state of affair. So here it's inhibited by RB. But once we have the, we have cyclin D and cyclin E pairing up with CDK4 and CDK6, those can come and can phosphorylate a retinoblastoma. And when it's phosphorylated, it is now inhibited, it's turned off. And you know what? That now means E2F is free to push us, yay, cell division, let's enter into S phase. But what if we were not ready to go into S phase? What if something's wrong? What if there's not enough growth signals or not enough nutrients, or we detect damage with the DNA? Well, then all of the P53s and the P proteins can come along and facilitate the CDK cycling complexes so that they do not phosphorylate E2F and it stays phosphorylate RB, so it stays inhibiting E2F so that we do not have cell division. So just to kind of summarize that, think of the P proteins as inhibiting division, the CDK cycling complex is prodivision, retinoblastoma as inhibiting division, and E2F as prodivision. And this kind of segues nicely into a tumor suppressor and a proto-oncogene. So some cells are anti-division and some cells are pro-division. You can think of these respectively as brakes and accelerators, right? When the accelerator's on, our car's going really fast, that is the cell cycle divide, going really quickly. Divide. And if the brakes are pressed, we're just not really going anywhere. We're not going to divide much. Now, if we have a defective brake, well, what happens if your car is a defective brake? You're going to go really, really fast because you can't slow down. So if we have a defective tumor suppressor, that could lead to cancer. Or if we had a hyperfunctional gene that's pro-division, that'd be like having the accelerator jammed on. That is also going to be pro-cancer. So we have tumor suppressors then and oncogenes. So very simplified, but we can think of as tumor suppressors as carrying less weight. So you just need one oncogene and it can lead to cancer. But again, super simplified. But one oncogene theoretically could lead to cancer, while you would usually want two tumor suppressors would have to be out of the equation to push towards cancer. This is known as the two-hit hypothesis. So we can kind of see this here. Imagine you have a car and there's, for whatever reason, a weird car, there's two brakes and two gas pedals. So what happens if just one of the brakes is fine? Well, if one of the brakes is fine, I'll just use this brake. I don't need that second brake. I kind of have a backup brake, which is nice. So you're good, no cancer, because you just typically need one brake. All right, what if both brakes are gone? Well, both brakes are gone, my car can't slow down, it's going to speed up, and that's more division. That's cancer. All right, what if I just have one of the gas pedals jammed in the on position? Well, if just one gas pedal is jammed in the on position, the car's going forward. It doesn't matter that I have a backup gas pedal. It's just irrelevant. Maybe even with the two brakes, the accelerator is jammed on, your car's probably going to go forward. So this would lead to cancer. Some people are born with one mutant tumor suppressor. So all of their cells have one of the brakes missing. And you can think of this as like flying without a co-pilot, right? If you have two pilots flying the plane and one of them passes out, well, you got a backup pilot, you're okay. But here we have no co-pilot. So you've got 40 trillion cells as an adult that divide, they're always dividing. It just takes one of those cells to get, so normally you're fine, right? But it just takes one of those cells to have a mutation. And you know what, now you have two tumor suppressors missing, and that particular cell can lead to cancer. The P proteins are therefore tumor suppressors. Here we have gas pedals should say oncogene. Oncogenes are the gas pedal jammed on, and another tumor suppressor, and again oncogene. P53, if this is defective, this is a tumor suppressor. If we have both of them out of commission, or if someone's born with a leaf foramen, right, they have one defective P53. Well, it's just dumb luck. Eventually, they're going to get another P53 mutation, and this is such an important tumor suppressor that as they grow up, they're going to develop cancers all throughout their body. Same thing with retinoblastoma. This is an important tumor suppressor that we just saw. And you're born, you don't have cancer, but it's just a matter of time. At a very early age, you're just dumb luck, usually in one of your eyes, your retina, hence the name, or your bone as well. So you should know that it's the bone and the retina. You might get another mutation there, and that could lead to cancer. You see something called leukocoria. So normally, if you shine a flash with a camera into somebody's eyes, you get something called the red reflex. So this is normal. You get kind of this like red reflection. But if you do it and you get leukocoria, leuko means white, well, that's just because you're hitting a big tumor and it refracts the light a little bit differently. Another simplified rule too, just didn't know where to put this, but if we're thinking about dominant mutations versus recessive mutations, dominant mutations typically are when you're looking at something that's structural. So here we're seeing collagen, fibrillin, myosins and sarcomeres. That's a structural thing. Huntington is not necessarily structural when it's wild type, but the whole point of the Huntington disease phenotype is that these proteins are going to start to aggregate, and this aggregation is a structural thing. So these are all dominant mutations. Recessive is, again, where we have this idea of a backup. So for albinism, tyrosinase is necessary to convert dopa to melanin. And if you have one defective, well, that's okay, you got a backup tyronase. But if you have two defective, this is now going to lead to albinism. Phenylketonuria, we need phenylalanine hydroxylase to convert phenylalanine to tyronase. But you know what? We got a backup. But if you have two missing, now you got this. Same thing for this with gangliosides. CFTR gene is getting colloid from one side of the cell to the other side of the cell. If we have a defect, that's okay. We got a backup to get chlorine across. But so this is where it's two missing. In hemoglobin and sickle cell, well, that's okay. We're just transporting oxygen. But if we have one defective, okay, well, we can still transport oxygen, okay, I guess. But if we have two defective, shit, now we're kind of screwed. This next section is focused mostly on epigenetics, so talking about modifications that we can make to the nuclear architecture to expose certain areas, making them more likely to be expressed, or to condense areas, making them more likely to be silenced. Looking specifically at heterochromatin and euchromatin, what Barr bodies are, how we can methylate and acetylate histones, not to be confused with the methylation of cytosines and what a CpG island is, and how all this plays a role in imprinting diseases. And then randomly, we're going to end it up with X-linked and trinucleotide repeat diseases, because I just didn't know exactly where else to throw all that. Your DNA can be bundled up and condensed into this large chromosome. Notice that it's kind of asymmetric, right? Above the centromere, it's a smaller half, and then below it's a larger half. So the smaller half is the P arm, and that's P for petite, and then Q is literally just the next letter in the alphabet. So the larger section is Q. So whenever we say chromatin is typically describing DNA plus the accessory proteins, typically the histones that are surrounding it. And a bit of DNA will have 8 histone proteins around them. So, we'll have 2 A's, 2 B's, 2 3's, and then 2 4's. And so that's why this is an octamer, and an octamer is really just a nucleosome. There is technically a 9th histone protein that links between nucleosomes. So again, 8 histone proteins with DNA wrapped around it is a nucleosome. We can have the chromatin sort of spread out. That's going to be euchromatin. Then we can have the chromatin condensed, and that is going to be heterochromatin. The heterochromatin is inactive, and the euchromatin is active. And the reason for that is, if you're trying to imagine fitting a transcription factor, it's got to bind to the DNA with the DNA, it's just got to bind to the DNA. It's a lot easier in the area where the DNA is exposed in the euchromatin. It'd be a lot harder for that transcription factor to fit in here. So the histone, we call them tails. So it's like the N terminus over here, it doesn't wrap as tightly necessarily. There's going to be a lot of lysines and arginines, and that should make sense. If the DNA is negative, we'd expect a lot of positive amino acids to be on histones. But modifications can be added to these tails. So like this lysine here could be methylated. And so if you're putting something on, it's a writer. You're taking it off, it's an eraser. And if you're reading a modification and doing XYZ function, then you're a reader protein. So this is just an example of a histone 3, the 9th lysine. It has 3 methyls, and then an eraser could take something off. Now it's 2 methyls, and then of course we could go back as well. So that would just be a methyl transferase and a demethyl transferase. And so pretty soon, though, we have 8 different histone proteins, all with different tails and lots of variations, lots of possible combinations of acetyl groups and phosphates and sumoyl groups and ubiquitins and methyl groups, and it's quite enormous and mind-boggling and complex code. We don't really have to know all of that. We can just simplify it to distill these 2 basic principles. If there's a methyl group, we're going to consider that group to be off, more likely to be in the heterochromatic state. And if there's an acetyl group, it is more likely to be on, spread out in the euchromatic state. And we can actually kind of make sense of the lysine. So if you were to imagine that the histone protein has a tail that, like we said, is a lot of positive things, it's more complicated than this, but we can imagine that it's kind of wrapping a little bit on the DNA because this is a positive, and so it's going to be very attracted. But if we add an acetyl group, because of resonance, this carbonyl carbon is going to be particularly positive, and so we wouldn't want positive there. So we've abrogated the charge. And if we get rid of the charge, now we can imagine that the arms are a little bit up. Now the DNA is a little bit more spread out, and the transcription factor could now bind. So the acetylation, we could actually make sense on a like very close up chemical level. But yeah, as I said, it's a really complex code, right? And the same way that AUG is a methionine, UAG is a stop codon, there is a complex epigenetic code. In addition to the readers that are reading all of this. And of course, we don't have to know this. I don't even know if scientists have fully worked this out yet. We probably need some complex machine learning thing to really crack the code. So girls have two XXs, XX, and gentlemen have one X. But you can't, I mean, if you think of like trisomy 21 Down syndrome, an extra 50% of the 21st chromosome can have really, catastrophic effects. So I know guys and girls are different, but you can't have two human sexes have such a dramatic difference in terms of having a whole extra chromosome. It's just not compatible. So there's got to be some way that we're making it so that people who are XX and people who have XY have a similar amount of gene transcription from that X chromosome. And so a couple possibilities is we could have more transcription in the male X, we could have less transcription in the female X, or, and this is what actually happens in our case, you can inactivate one of these X's. And these are called bar bodies. So here's a zygote, here's two functional X's, and then pretty early on during embryology as, we're forming a blastula and a gastrula and whatnot, some of the one X chromosome or the other will inactivate. And it's actually quite random, and it's not always the same one. So we end up with this complex pattern, this sort of mosaic of, X from mom compressed versus X from dad compressed. And to compress them, there's just a hyper heterochromatic state. So it's a lot of methyl groups and not a lot of acetyl groups. You could use bar bodies in this mosaic model to explain the tortoiseshell color on cat. So if we look at this cat, and we see this tan fur and this black fur, we can explain this using bar bodies. So just imagine that the purple X that we see here is for tan fur, and this other X here that's teal, it's going to be associated down the road with cells that produce the protein for black fur. And again, we're going to have these just completely random inactivations, and so this one's got its descendants will express the black color, and this one's descendants will express the tan color. And so if we kind of play that out and continue playing that out, we're going to get little pockets where some areas are black and some areas are tan. So we methylate the histone protein, but you can also methylate the cytosine. And it actually works out a little bit easy though, because in both cases, it's going to lead to condensation, repression, silencing. So this would be methylated DNA. It's typically going to be associated with compression. A CPG island. So this is the term. So term CPG, C is just cytosine, and then G is just guanine. So what's this P here? Well, nucleotides are linked via phosphate between the 5' carbon of one sugar and the 3' carbon of the nucleotide sugar above it. So P is just for phosphate. It's kind of stupid. We could have just called them a CG, and I think it would have made just as much sense. But that's what I mean. So it's an area in the promoter where you might have a lot of methylation. And if you have a lot of methylation, this area is going to be turned off and condensed. Imprinting. So babies are little parasites. Why? Because they're sucking all the nutrients from mom. And problematically, sometimes they suck a little bit more nutrients than is healthy from mom, right? So let's think about incentive. So if you imagine, a couple trying to sell a house and their realtor, it seems like their incentives are aligned, right? If you sell a $300,000 house, it's a 6% fee, and half that goes to the buyer, half that goes to the agent. So at the end of the day, a $300,000 house, the agent gets $4,500, right? So the agent is incentivized, you would think, to try to make as good a sale as possible. If instead of selling the house for $300,000, we sell it for $600,000, well, then the agent makes $9,000. So obviously, again, the agent and the people selling both want to sell it for as much as possible. But if you think about it a little bit more, maybe they're not completely aligned. Because 300,000 is 600,000. That's easy if it's just if it's just one deal or the other. But imagine just to get an extra $10,000 for the house. It's more flyers that we got to pull up. It's longer. We have to leave it on the market. It's more calls that the realtor has to yield. It's 30% more work. It's like an extra month or two of her time to get the house for $310,000. Well, the couple definitely wants that extra $9,400 after the 6% fee goes to the realtors. But what's actually getting in the pocket of the realtor who's selling it? Just $150. After the realtor's agent, 50% is going to her broker fee. So in case the math didn't add up for you. So 30% more. She's only making an extra $150. It's just not a good use of her time. And so she actually might want to sell the house for cheaper than they do. So why am I telling this silly story? Because it kind of explains imprinting and the mom and dad's genes. They both want the baby to be as healthy as possible, right? They both want it to be healthy and fit. They want the mom to be healthy and fit because she's going to help her other child, and they both want the baby to be healthy and fit. So it seems just like the realtor and the people selling the house, there's an alignment of incentives. But the dad's genes actually, is a little bit, a fair amount less investment of resources, his ability to generate another child is fairly easy. So it might just be this tiny little crumb more in favor of stealing nutrients from mom at her detriment to the baby's benefit. And so we actually have, this misalignment of incentive. This is really the entire idea behind imprinting. So look at these two genes. These are the two real genes, the insulin growth factor 2 and H19. So insulin growth factor's job is to allow more sugar into the cell, which allows the baby to grow more. And then H19 is going to inhibit growth. And so what we see now is that for this top picture here above this line, this is somebody, a male or a female, and they are choosing to methylate, in other words, imprint this H19 gene, and they're letting the IGF-2 be normal. And then it's vice versa for the one below this line. So if you had a guess that the top chromosome is a male or a female, it would make sense that it's a male and the bottom one's a female. Because in this case, we're going to, this is on, we're going to steal more nutrients, and this is off, the inhibition of growing. And so that's exactly what we get. So if we look at another gene, P57, so P57 is going to be a CDK inhibitor. CDK is pro-growth. And so males are going to want to inhibit the inhibitor of the CDK, which is for growth. And the females are going to want to have it active because they don't want the baby to grow so much. This is the male right here. A male has one chromosome from dad. And again, the males typically want to methylate this area. So it is the paternal chromosome is methylated. The maternal chromosome, it makes sense, does not want to methylate it. She wants that open. Now, when this man generates sperm, whether the sperm comes from this chromosome or whether it comes from this chromosome during meiosis, it doesn't matter. We're going to erase all methylation programs and reset them to the male preference. So all of the male sperm, regardless of which chromosome, now have a methylation of this particular gene. And females would be the exact opposite. So again, females, though, they have one copy from dad and one copy from mom, and the dad copy, just like in the gentleman up here, is going to be methylated, and the female is not. But again, whether it's this chromosome going through or this chromosome going through, we're going to erase whatever the setting is and reset it. And the woman would like to have this gene expressed. She doesn't want the baby to grow so much inside of her and steal a lot of her nutrients. So an idea which synergizes with this relatively well is uniparental disomy. That's probably a new term to you, but you guys probably learned in undergrad the concept of non-disjunction. So during meiosis, we are going to separate the homologous chromosomes first, and then we are going to separate the sister chromatids. And if we look over here, this actually should say 2, and this should say 1. So if we look over here, everything split fine here, but we have a non-disjunction of it in meiosis 2. So we have too many chromosomes here and not enough chromosomes here. This is something that would lead to like Down syndrome, trisomy 21. And in this case over here, non-disjunction of it in meiosis 1, so that we have too many chromosomes over here and not enough over here. So that's non-disjunction. You guys know that, but what is uniparental disomy? Imagine that this, let's just say this is an egg. This egg goes up here. Excuse me. Let's say this is chromosome 17. So, the fusion happens, we get a zygote. There are now 3 copies of chromosome 17. Sometimes this will be bad and you'll get something like Down syndrome, or maybe it'll just be a spontaneous abortion. But sometimes somehow the cell can decide that it should destroy one of these chromosomes. And if it destroys this chromosome over here, we get this, which is really just maternal disomy, which means both of chromosome 17 are from mom, right? Normally everyone's chromosome 17, one from mom, one from dad. Here we have 2 from mom. Okay, well, what if we have this other situation go over here where there's just no chromosome 17. Now I only have one chromosome 17, and that's about as bad as having too many copies of chromosome 17. Well, again, the cell can sometimes like recognize like something's a little bit off, and it can duplicate this chromosome. So now we have a paternal disomy. So uniparental disomy is when you have a non-disjunction event, and then there's a kind of rescue, but you don't have heterogeneity to the chromosomes. This can, you could also have mutations and deletions, but I would think that this is going to be a major cause for something like imprinting. And if this occurs, imprinting typically is we're going to have mom version is off and dad version is on, or dad version is off and mom version is on. But if I have this uniparental disomy, I might have too much, both versions on, or both versions off. So let's look at the P57. Dad's is off, mom's is on, and it might turn out that just one dose of P57 is like ideal or standard, at least as the baby is an adult. If in this case we had a uniparental disomy from the male, we now have two copies of the sort of male imprinting pattern, the silencing. So we're silencing this inhibitor of growth, so we're going to have too much growth. We look at IGF-2. You could kind of have the same thing over here. So I guess this would be another male disomy. We have two, this is pro-growth. So here we have inhibition of two genes that are inhibiting growth, and here we have activation of two genes that are pro-growth. This is just too much growth. So both of these would lead to the same phenotypes of macrosomia, where the baby is too large because it's sucking too many nutrients from mom. And it's not just sucking nutrients from mom. The cells are all about taking nutrients from their surroundings and growing and dividing and growing and dividing. What does that sound like? That's cancer. And so Wilms tumor is a common renal cancer that kids get due to imprinting issues, sometimes due to mutations, deletions, often due to uniparental disomy. So Prader-Willi and Engelmann is a similar thing as well. These are more imprinting diseases. So turns out that on one long stretch of chromosome 15, we have a region, the actual genes don't matter. There's a couple genes, let's call them W, X, Y, and Z. So these are on in the male. So we would probably assume that these are somehow pro-growth, but let's just drop that point. They are turned off in the mom. They are silenced, they are imprinted, they are methylated. And then a little bit down on this chromosome, we have these two more genes, UB3 and A. UB3 you actually do have to know. So UB3 is the only one that's real you have to know. These, conversely, are silenced, methylated, repressed in the males, and expressed in the mom. So if we go ahead and look at the wild type over here, on the maternal chromosome, we have W, X, Y, and Z imprinted, which means turned off. The only genes you have that mom's bringing to the table are UB3 and A. And on the male, all the males bringing to the table is W, X, Y, and Z. A and UB3 are imprinted. That's normal. So what's happening is we're getting one functional copy, one copy of UB3 and A from mom and one copy of W, X, Y, and Z from dad. If we look at the Prader-Willi syndrome over here, so the mom chromosome is fine. UB3 and A are there and just that's normal. But on the paternal chromosome, there's been a deletion. And if there's been a deletion, then we do not get the W, X, Y, and Z. Because remember, it's typically dad's job to bring W, X, Y, Z to the table, but we've had a deletion there. So this deletion is responsible for Prader-Willi. And I always thought of like P for paternal, paternal. If you look at Engelmann, it's the opposite. We have its deletion on mom, and so the paternal copy is fine. So we don't have UB3 or A, so the maternal deletion is now responsible for this. So again, W, X, Y, and Z and A, or you don't have to know. The only thing I would know is that UB3 is on the mom, and paternal deletion is associated with Prader-Willi, and maternal deletion is associated with Engelmann. So what are the phenotypes? Prader-Willi, hyperphagia, and obesity, intellectual disability, hypogonadism, and hypotonia. So there's not much muscle development. For Engelmann, seizures, ataxia, intellectual disability, and this very particular type of laugh is described as a happy puppet, which is kind of a weird way to describe it. Moving on to X-linked symptoms, ornithine transcarbamylase, Fabry, glucose 6-phosphatase, Hunter. We've already talked a bit about these, so we're going to focus on these over here. Turner's, Klinefelter's, Rett, muscular dystrophies. Note that the last one is not X-linked. I just included it because I thought it was a good idea to bundle all the muscular dystrophies together. And then Fragile X, which is a trinucleotide repeat. So Turner and Klinefelter. So this has to do with non-disjunction of the sex chromosomes. Klinefelter is XXY, and then Turner syndrome is X not. So there's not a second X chromosome, it's just one X. These are for the Klinefelters, and these are for Turner syndrome. For Turner syndrome, I would say the web neck and the shield chest are going to be real big standouts in a vignette. Now, this is a lot of characteristics. I think if you just think about forget about our bodies for a second. Just assume, and it kind of makes sense, that the X chromosome might have some genes that have to do with female secondary sexual characteristics. And so for Klinefelter, if we have extra for the X, then we're gonna. So imagine that X has the height gene and sexual characteristic for females. So very tall makes sense. Not a lot of beard growth if you have less testosterone and more estrogen. Breast development because you have estrogen, X chromosome. Female pubic hair pattern, again, you're going to have estrogen, not as much testosterone, more estrogen, testicular atrophy. And the short stature, again, we're just pretending that the X chromosome has an important gene for height, which maybe it does. If you don't have you're not going to have as much estrogen, your ovaries won't develop, and you'll have amenorrhea, and you also need your ovaries to produce estrogen. And so you have underdeveloped breast. Estrogen promotes and contributes to breast growth. And then you have these little moles randomly on the body. That one's, I don't really know. Rett syndrome, so X dominant. However, 95% of cases of Rett syndrome are just de novo mutations during gestation. And the protein here that's mutated is MECP2, and this stands for methyl CpG binding protein. And so we said that CpG islands are just, CG in the promoter. They're often hypermethylated and they get turned off. They get condensed. Well, this is actually the protein that binds. It spots all of these methyls in the promoter. It binds them. It's actually that does the condensing. The condensing. Now, in Rett syndrome, this is dysfunctional, so you don't actually get silencing. And it turns out if boys get this, it's just going to kill them. You need to bind the CpG islands and condense them. Now, in these females, this is just a very important thing to do. So in females, they have a backup, but I guess it's not enough to fully rescue the phenotype. But boys, it's just, if you don't have any condensing of the CpG islands, then you just won't. It's an embryonic lethal. Severe motor and cognitive dysfunction will develop. Muscular dystrophy. So dystrophy just means that the muscles are going to waste away. So we have three different kinds: Duchenne's, Becker's, and myotonic muscular dystrophy. Becker and Duchenne, notice they're going to be more similar. They're both X-linked, and the myotonic is actually quite different, autosomal. So for Duchenne's, we have this dystrophin protein, which is just important for muscular stability and development. This dystrophin protein is an incredibly large gene. It's one of the largest genes in the human body. And that's why there is just a large likelihood of having a mutation here. Often they're frame shift mutations, a deletion specifically, and this might result in a truncated non-functional protein, which will contrast with Becker in a second. So it's such a large gene, you're going to have a mutation somewhere, and if it induces a frame shift, you get a shitty protein that doesn't work. Gower's sign is this very specific, because they have muscle wasting issues and not a lot of strength, to if they're on the floor, to stand up, they will stand up in this very characteristic way that they might describe in the vignette. And again, very severe phenotype, so the muscle weakness will be very young. Becker is almost the exact same thing, dystrophin protein, frame shift, but it's a functional protein. It can still do its job, just not as well. So they say that Becker is better, is a way to think of it. And it's early adulthood instead of, right when you're born, because again, the phenotype is less severe, and that makes sense because the protein's working a little bit. Myotonic dystrophy is for the DMPK gene, dystrophia myotonin protein kinase. It causes myotonia, where you can't actually, if you contract your muscles, it's hard to relax them. So the example that's often given is that if you do a handshake, it's just hard to let go, basically. People develop cataracts. There's hormonal issues. So testicular atrophy and premature balding. And this is a trinucleotide repeat. It's one of our 4 trinucleotide repeat diseases. The second trinucleotide repeat disease is Fragile X syndrome. So this is the most common the most common inherited cause of intellectual disabilities. Down syndrome is actually more common, but it is sporadic, so it doesn't count as an inherited disease. And FMR1 is the gene that we're talking about, and it stands for Fragile Mental Retardation 1 gene, which kind of makes sense. Trinucleotide repeat, it's a CGG, and if we make another link to CPG, that those islands, they often, for whatever reason, get spotted to be methylated. So this is not a promoter. This is not a CPG island, but because we have so many CG, some system, some protein in the cell sees this and might think it's a CPG island, and it hypermethylates it, which can turn this gene off. Large testes, that actually comes up in vignettes, is a thing. Large jaws, and particularly large ears, which we can see here. This kid is just absolutely adorable. Trinucleotide repeat diseases. Huntington's is going to be really common, highly tested. We've done Fragile X and myotonic, so just Huntington's and Friedreich. For all of these, the more repeats you have, the more severe the phenotype is. So this is for Huntington's and CAG repeats. And so it's not just the more repeats you have, it also happens to be the case that typically as you accumulate repeats, it's easier for your body to produce more repeats. So over the generations, the grandpa might have 40 CAG repeats and have mild Huntington's, and then their kid will have 52 repeats and have moderate Huntington's, and their kid will have 74 CAG repeats and have a severe phenotype. That would also explain that if your phenotype is more severe, for the grandfather, the Huntington's hit them in adulthood, for the parent a little bit earlier, and for the kid with so many CAG repeats, not only is it very severe, but it hits them very early. These terms are called anticipation. Friedreich ataxia is the GAA repeat of the frataxin gene, which is important for iron regulation of the mitochondria. It's going to present quite early. You're going to have kyphoscoliosis. So this is where you have both sagittal and coronal displacement of the spine. Ataxia, hence the name, so balance and coordination issues. Diabetes, hypertrophic cardiomyopathy, so it might be like a young person, the heart basically gets too big and you get a heart attack. And so people and athletes in their early 20s might just die, basically. And I guess if you have Friedreich ataxia, we know the phenotype, but they might just describe somebody who has a heart attack, and that's hypertrophic cardiomyopathy, and it's associated with this. Pes cavus is this super arched foot, and the hammer toe is not this big toe, but these other toes are the hammer toes. Huntington's chorea, sometimes it's called, it causes these involuntary movements, and that's because the basal ganglia, the basal ganglia has 2 different pathways, a direct one, which is like to initiate movement, and indirect one to inhibit movement. The indirect pathway is destroyed in the basal ganglia. So all of the movements that your body typically wants to do, but you don't even know about because your basal ganglia says, no, let's not do that movement. It'd be weird. It's now not working here. So they get this chorea phenotype presentation. It also affects their mental state, their personality, depression, aggression, and behavioral changes, and eventually leads to dementia, and hits people particularly early as well, in 30 or 35. DNA replication. All right, we're going to start off just really hammering home exactly what 5' and 3' mean, and a couple different things they can mean. We're going to go through the entire mechanism, and we're briefly going to talk about some, drugs. All right, so here's carbon number 1, carbon number 2, carbon number 3, carbon number 4, and carbon number 5. Carbon number 5 can be attached to a phosphate, and carbon number 3 can be attached to a phosphate. If we, look at this nucleotide A, B, C, and D, right? This is a tetranucleotide structure. We see that for D, 3' is not really bonded to anything, not to any other nucleotides. And here we see that for 5, or nucleotide A, it's the 5' that's not really connected to anything. That is why this is the 5' side, and this is the 3' side. Again, we have a 5' end and a 3' end, and now they run anti-parallel. So the thing that they're attached to, the 5' end will be on the bottom and the 3' end will be at the top. So to really super duper hammer it in, think of like Georgia and Florida, right? So here we have Florida, here we have Georgia, and here we have New York City. So Georgia is north of Florida, and yet it is south compared to New York, right? So something can be north and south at the same time. Similarly, if we look at a 5' and a 3', and again, all this like the absolute like North Pole, what this 5' means is just that somewhere here we have a 5' carbon on a ribose that's not attached to another nucleotide, and here we have a 3' the 3 carbon on a ribose that's not attached to a nucleotide. That's where we get the absolute north and south pole. But again, things can be relative. If you pick this little position here with respect to this end, we can call this the 3', and with respect to this end, we can call this the 5'. So it's very important, I think, to understand that relative 5', 3' directionality. So whole mechanism. Helicase shows up, starts unwinding. Boom. Look at that. We're going to watch that again. That is some cool ass graphics. All right. So now we have these single-stranded binding proteins. So DNA is negative, so you can assume that these are super positive. And why do they bind here? It's because if they don't bind, everything, these things are going to try to start, becoming a helix again, right? They want to reanneal, and we don't want that to happen. So the SSBs prevent reannealing. Primase is going to go ahead. Now, anytime, there's only one thing I can think of that goes 3' to 5'. And so pretty much when in doubt, things are always laying down track, always laying down RNA or DNA in the 5' to 3' direction. So notice if this is 5', then I would say that this is 3', right? 3' and 5'. And so what the primase is going to be laying down is 5' to what we would call the 3' direction. And that's exactly what we see. It's going to lay down in the 5' to 3' direction. All right. I always thought of like DNA polymerase as being kind of a diva. It's like it needs a red carpet to start off, right? I won't even start doing my job unless I can walk off of a red carpet. And RNA is often depicted as red. So I'm seeing the DNA polymerase only starting at the end of the red carpet, the red RNA primer. This is going to be really important later for things like telomeres. So DNA polymerase is going to extend 5' to 3' in that manner. As helicases, we can see moving to the right. So it's DNA polymerase 3 in bacteria. It's epsilon in eukaryotes, and it's so we have a leading strand and a lagging strand, which we'll get to. I always thought of like Delta Airlines. You might have like a jet lag, or that's how I remembered it. So the helicase is moving to the right, and the strand that's being the new strand that's being synthesized is moving in the 5' to 3' direction. So in other words, it's moving down here. It's sort of chasing the helicase. It's moving in the smart direction, right? That means it can just continuously run chasing the helicase. We're going to have the lagging strand, which is going this 5' to 3' direction. It's going to be moving in the opposite direction. So it's kind of stupid. It has to keep stopping and starting, stopping and starting. So let's see exactly what I mean. Smart leading, boom. Look at the stupid. The stupid has to get up, run all the way around, get another primase, lay down, and do that again. It constantly has to keep doing that. It's just a little bit silly. So this is just a much better picture of the leading and lagging strand. The DNA is unzipping because the helicase in this direction. So this can just follow it forever and ever and ever without having to pick up. But this guy constantly has got to go, oh, crap, oh, crap, oh, crap. Oh, and it's just like a real hassle. Notice as well that every single one of these replication events is starting off of a little red RNA primer right there, etc. This is a bubble. This is going in both directions. We've only been depicting one side of this, where we have a leading strand here and then a lagging strand on the other side. But one man's lagging as another man's leading, right? So on this side, we have another helicase going this way, and a helicase going this way. And so now this is actually being the smart one on this side, chasing the helicase. So it depends. On one side, you're the leading, one side, you're the lagging. Okay, so here we have Okazaki fragments. So we had an RNA primer. So the DNA polymerase, which made this right here, it can't go, it's got to stop when it gets to the primer. So this is a little chunk, and this is a little chunk. and those are our Okazaki fragments. Now RNAs, so Okazaki fragments, RNAs can now cleave the RNA, kaboom, get out of there. And now DNA polymerase 1. So it's important. DNA polymerase 1 is replacing the Okazaki fragments. Polymerase 3 is laying the main track. This can just go ahead and moves 5' to 3' like everything. And it can fill in most everything. But notice we still have these little gaps, right? So DNA ligase is going to come in here and close those gaps. SSBs were here the whole time. It just was too lazy to draw everything. So they're just always there trying to prevent reannealing. Ahead of the helicase, we have topoisomerase, or called gyrase in bacteria. It relieves tension. What tension? Well, if you have a lanyard, or you have like a bit of string that you can twist up into a double helix, twist it up into a double helix like a smia, and then just start to like unpeel it like this. And what you're going to see is you're going to induce all of these tensions and coils. And if you do it too much and it's fragile, it might break. So the idea is that if you had topoisomerase, you would keep it nice and steady and prevent all of this. So there's two types of topoisomerase 1, topoisomerase 2. Number 1 cuts, you can see this little itty baby 1 cut right there. And here I can see that I've cut once and I've cut twice on both sides. So that makes sense. The 2 cuts the 2 strands. And names you should recognize are toposide, teniposide. So I always thought of like there's 2 sides to every story. And then Tekken, I always thought of Tekken as like the #1 video game of my childhood. So Tekken, the stupid fighting game from the 90s, is Topo 1, silly. Because they're inhibiting topoisomerase, specifically in eukaryotes, these can be used as chemotherapies. So cancer cells dividing too much, let's fuck up its ability to relieve supercoils and tensions when it's dividing, which it does a lot of. You also have fluoroquinolones, they're like ciprofloxacin, and these can inhibit bacterial gyrase. So these are going to be antibiotics. This is just a nice summary. So we're going to lay down a 5' to 3' primer. Boom, we have it right there. We're going to extend with DNA poly 3, which is going to chase the helicase in this direction. Here we have topoisomerase. This is the leading strand. It's chasing the helicase. But then we also have primer here. And then we have DNA poly 3 and another one, DNA poly 3. And that's why we have these 3 Okazaki fragments. But eventually we don't want the RNA. So then we have we're gonna get rid of it with RNA. So we're gonna try to fill in the things with DNA poly 1, and then we're going to ligate those Okazaki fragments together with DNA ligase. And this is all happening on one side. The leading and lagging will flip on the other side of the replication bubble, as we see there. All right, so full disclaimer, you guys totally need to know about telomeres. Having said that, I probably go a little bit too deep into telomeres just because I think they're super cool. So just go into this section knowing that. And they're involved in cancer and aging. They're fascinating. 3 things that we'll talk about. 1, why are they wrapped in like a little loop like this? And then why do they have all of these proteins sheltering them? 2, why do they get shorter over time? And why is this TTAGGGG repeat necessary then? Or why is that what we find at the end of the chromosome? What's the purpose there? 3, if telomeres are shortening, how do cells take advantage of this? And what is the Hayflick limit? So why are they tucked in this little loop here? And then why do they have all of these sheltering proteins? One of the worst things that can happen for a cell is that we get a double-stranded break. So boom, we got a double-stranded break here. Alarms go off. A bunch of repair mechanisms are alerted to this open exposed end, and they want to fix it. Well, this exposed end right here to the cell doesn't look anything different than what we see right there, right? So the same way a cell would try to repair a double-strand break by stitching it together with something, it's trying to stitch telomeres together as well. We don't want that. We don't want them to be stitched together, because of course they're not broken. They're just the end. So again, a cell cannot tell the difference between a double-strand break and the end of a telomere, because if you think about it, they kind of look like the same thing. So the solution is let's wrap them up and let's put all these proteins on them. just to really underscore this, if you do a knockout of one of those sheltering proteins, you get this. You get telomeres, which are all stitched together, which, as you can imagine, makes mitosis and anaphase like a real bitch to deal with. It's just really difficult to separate everything in the way that we need to. This next thing, definitely not on the exam. I just thought this was cool. Proteins sometimes moonlight, like acetonase is going to isomerize citrate to isocitrate, but it also plays a role in like iron regulation. Similarly, these proteins, these sheltering proteins, are just supposed to like protect and shelter the telomere so that it inhibits double-strand break repairs. But this RAP1, one of these proteins, can dislodge, move over, and act as like an activator of transcription for metabolic genes. And that's just, to me, that's really interesting. And as the telomere gets shorter over time, there's less shelter in proteins because there's less shelter in proteins on the telomeres, so more of them are dislodged, so you get more and more of this RAP1 going over. This might be one of the reasons that people gain weight as they get older. I'm sure it's a lot more complicated than that. All right, so why do telomeres shorten, and what's the point of the TTA-GG repeat? So first, just fact, we do have a bunch of TTA-GGG repeats, like 10,000 of them. And it's often been described as an aglet, as a little protective tip of a shoelace. And so we'll see in a bit why that's the case. And we might even ask the question then, like, why do we need this protective TTA-GGG if we already explained why it loops and has these sheltering proteins as protection? And it is all about replication. When it replicates, it is, even one, it's going to unwind, this is going to straighten out, and two, it's going to lose the sheltering protein. So it's when it divides, it loses a little bit of information every time. So why is that the case? Why are we losing information? It's called the end replication problem. We saw this earlier. Prime is going to lay down a primer, 5' to 3', okay? DNA polymerase is going to extend, okay? And then we're going to get rid of the primers, just like we've been doing, right? And now, ideally, what we'd want to do is fill in these spots with DNA, right? You always want to fill in the RNA with the DNA after the RNase gets rid of it. But the problem is that you need to extend DNA polymerase, right? It's got to walk off the red carpet off of an RNA primer. And we don't have anywhere to put a primer. So if I can't put a primer here, right? If I can't put a primer there, then I can't get a DNA polymerase to extend off of it to fill in that gap. So this is what happens. We replicate. We cannot fill in these gaps that we see there. And so this overhang is naturally just going to be dissolved. So again, that overhang is just naturally dissolved. And over time, really, these are the sections that are going to get copied. And we've lost information on the end, and it's just going to progressively get shorter and shorter and shorter. We probably lose about 100 base pairs per division. So that's why there's just an intrinsic limitation to the polymerase machinery that it must move 5' to 3', and it must extend off of an RNA primer. So if I'm going to lose important information off the end of my chromosomes, the linear chromosomes, well, I don't, if this is my insulin gene right here at the end of the chromosome, I sure don't want to lose my insulin gene or parts of it. So what happens is that the telomeres, that basically having a TTA-GG repeat is useless. So it doesn't actually matter if I start eroding away at the TTA-GG repeat. We can afford to lose this because it's just kind of useless, repetitive information. So that's one of the theories, one of the reasons why we just have this useless, repetitive information rather than important genes. If you had an important gene, that'd be kind of like a recipe for disaster. So then the last interesting thing is, so we talked about why they shorten and why we might want a TTA-GG, like useless information instead of like an important gene, protein coding sequence. But what is the Hayflick limit? So to understand the Hayflick limit, it's just seems obvious if you just think for a second that as longer a cell lives, the more mutations it's going to make, the more changes to the epigenome, the more protein garbage is going to accumulate. The cell's just going to become kind of shittier, for lack of a better word, over time. This decreases the functionality and increases the risk that these are going to become cancerous. So old cells are dangerous cells. They don't function and they're likely to become a cancer. So because the telomeres shorten at such predictive intervals, the cell can then use them as a timer to realize how long they've been alive. So rather than think of a prisoner as like putting tally marks for how long they've been in prison, it's as if they arrived in prison and there was like 10,000 tally marks, and then they start erasing tally marks. They erase 100 every day or so, and then they could use that to infer how long they've been there. It's the same thing for the cell. When their telomeres get short enough, when they've erased enough of the tally marks, they go, okay, we've probably been here long enough. It's probably time to do cellular suicide. So just to see how many analogies I can squeeze in here, you could think of a telomere as a wick for self-destruction. So we have a certain amount of TTAG repeats. In every division, we get rid of a few, we get rid of a few, we get rid of a few, and boom, when it gets too short, this cellular senescence, this apoptosis event, actually probably more cellular senescence than apoptosis, the cell is just going to, basically take itself out of the equation. So if we're going to start with a telomere that's long, we have 10,000 base pairs, days in culture, and the number of populations doubling. So what we're seeing is the cells are dividing. The cells are dividing over time, The telomeres are getting shorter. And then it starts to plateau. We're getting less division, less division, less division, less division. And that's because the telomeres are getting short. They get short enough that these cells just stop dividing, become senescent. And so this is the Hayflick limit. It's how many times a cell can divide before it just sort of stops. And the reason it's stopping is the telomeres are getting short. And it's going to vary depending on if you're talking about a fibroblast or an adipocyte or whatever. Fibroblasts are like 50 something, and it's going to vary. So what if you could extend the telomeres, right? Could you outsmart the self-destruct clock? Yes, there's a way to do this. It's called telomerase. So telomerase is going to extend one of the issue was, oh crap, we can't lay down an RNA primer to extend off of, but what if we had more runway so that we could make an RNA primer? So this is the replication that we saw earlier. And now what happens is telomerase comes into the picture and gives us more of that. So again, here's telomerase, and it's extending so that now I can put an RNA primer, and then my DNA poly 3 can move, 5' to 3'. Or we could think of 5' to 3'. It's all relative. So we can extend now off of that in the 5' to 3'. And boom, now I've maintained the original length of everything thanks to telomerase. So some cells do this. Embryonic cells do this because the germline, needs to have, replicative immortality, right? I have a grandson and he has a granddaughter, and et cetera, et cetera, et cetera. We're not going to slowly age as like a carcinous population. The embryonic stem cells stay young. Adult stem cells stay young a little bit. They have some telomerase, but not as much telomerase. And if you look at a somatic cell, like a normal body cell that's getting old and getting ready to die as the number of divisions is increasing, what's happening is at B is we're activating telomerase. We activate a lot of telomerase, and now the cell can just divide forever. And typically it's going to become a cancer cell. Pretty much all cancer cells have telomerase upregulated, and that's so they've turned off the self-destruct timer. Again, I told you we're going to cram a lot of metaphors in here. So you can think of the Hayflick limit is like a term limit, right? We don't want politicians in office for forever because we don't want cronyism to develop, and we want to get fresh blood in there. And it's the same for cells. That's why we have these inbuilt timers, so we can say, all right, these cells can get out, and let's bring some new cells in there. All right, DNA damage. We're going to talk about the different types of point mutations, how point mutations varies from a frameshift mutation, and then the types of frameshift mutations that we can have. There are basically like 4 categories of DNA damage, and therefore there's like 4 categories of DNA damage repair: base excision repair, nucleotide excision repair, mismatch repair, and then double-strand break repair. All right, so let's look at our wild type over here. So this is our DNA codon, and then this is the RNA codon. Now we have to ask ourselves, are we looking at the coding strand for the DNA or the non-coding strand? This must be the non-coding strand. In other words, the template strand that they've shown us here in this illustration. Because if it was the coding strand, it would have to match. So that's always something you should immediately jump to if you see something like this. Happens to be a lysine. Okay, point mutation is where we just change one of these nucleotides. All right, if you change the third nucleotide, about half the time, it won't matter. It'll give you the exact same amino acid. This is because of something called wobble that we'll talk about later. But so if the third nucleotide often doesn't matter, and when it's the same amino acid, that's silent. A nonsense mutation. So we have UAA, UROA. UGA, you go away, UAG, you are gone, right? These are our 3 stop codons. So if we convert a normal amino acid into a stop codon, that is nonsense. We get a truncated protein. If you truncate it on the 5' end, you're going to have an even shorter protein. If you truncate it on the 3' end, you're going to have a little bit of a longer protein. Missense means that we changed one nucleotide and it did actually change from one amino acid to another. Within this, we have conservative, where lysine is positive and arginine is positive, and the non-conservative, where it's just not positive. It's threonine, so it's not conservative. I guess you could think of this as a spectrum, however, where we'd have something like an arginine would be on the conservative side, and then the non-conservative side. Maybe here we have something like threonine, and maybe here you have something like, glutamate or aspartate, because that's negative. So that'd be even, so it's not a binary. It could be varying degrees of conservative and not conservative. So original, we just went through all of the point mutations. Now a frame shift mutation. We are now adding a nucleotide or taking away a nucleotide and shifting the entire reading frame. Keep in mind that this, if you do it of multiples of 3, it will actually not be a frame shift. So 3, 6, 9, 12 wouldn't be. So, but if it's 2 or 4 or 5 or 7, then yeah, you're good. And this is going to be about as destructive as a nonsense mutation. Again, if it's on the 5' side, it's going to have more, like a larger effect than if you did it on the 3' end, because in that case, you're only screwing up the last couple amino acids. And yeah, this is pretty much what we just saw. We're shifting everything out of alignment, right? It should be CAT, CAT, CAT. And just adding one thing, now the CAT, CAT is not read in the proper frame. We have a new reading frame, and so everything is screwed up. DNA polymerase is pretty good, makes 1 in 1,000 mistakes. It has a proofreading domain, which is pretty much the only thing I can think of that moves in the 3' to 5' direction. This is also pretty good, 1 in 1,000 mistakes. And then we have mismatch repair to see if we screwed up and put like an A with a G or something like that. That's also pretty good, 1 in 1,000 mistakes. 1 in 1,000 times 1 in 1,000 times 1 in 1,000 is 1 in a billion. So we're only going to make 1 in a billion mistakes, which might seem great until you keep in mind that we have 40 trillion cells, which are dividing a lot. And each cell has 3 billion nucleotides in it. 6 billion if you can count the fact that it's diploid. So that means every time we divide a cell, we might make 6 mistakes. And over 40 trillion cells, no wonder earlier when we saw someone has like retinoblastoma, flying without a copilot, you're bound to get a mutation in one of those cells. So so it seems like pretty good, and it is, but it also leaves a lot of mutations to accrue. When you're loading a nucleotide, you always load a triphosphate. And then as it loads, you're going to pop off a pyrophosphate, 2 phosphates. And we are moving in the 5' to 3' direction, as everything does. Except for our, we've now incorporated a GG. Wait a second, GG, you don't belong. So this exonuclease domain, not really shown, but mentioned, is going to read it in the 3' to 5' direction. And because it's not a match, it boots it out. And then we can keep moving. Base excision repair is for just one tiny little nucleotide of a difference. So it's abasic. We're either missing the nitrogen base attached to the deoxyribose, or there's been a deamination, like cytosine to uracil or 5-methylcytosine to thymine, something like that. So I do want to make a comment. Base excision repair sometimes sounds like mismatch repair because we might have nucleotides that aren't properly matched to one another. But the difference is in base excision repair, we know who's the imposter typically because it's a nucleotide that's not a canonical one. And so it's easy to spot the imposter. Nucleotide excision repair, if you've got like a kink in your DNA. So if you have pyrimidine dimers because you have been in the sun too much, or you have a bulky attic because you've been in the lab and you splash some ethidium bromide in your face, that's going to be NER. Mismatch repair is if we make a replication mistake and we don't catch it with our exonuclease. And then double strand break repair can happen for a variety of reasons. We have 2 repair mechanisms, HR, also called HDR, which is reliable, and then NHEJ, which is we just need to patch this up as quick as possible. We don't care if it's an accurate recreation of the original strand. So again, we can see a nice picture of all of these. The ones that you really need to know, the genes, is the MSH and MLH has to do with Lynch syndrome. And then BRCA has to do with breast cancer, ovarian cancer. And then ATM will be a telangiectasia ataxia, which we'll talk about. So BER, look here, we're just missing a little base. And so what we do is we cleave with an endonuclease. So endonucleases are going to cleave like internal, like on the inside of a long DNA strand. Exonucleases will come in from the end. So, exonuclease would, come from here, and an endonuclease would just start right in the middle. So, we're missing a base. Endonuclease then just cuts out in the middle, and then we just have a polymerase fill it up and ligate it. Now, I would actually memorize that right now, what I just said, because that's going to actually follow for pretty much every single type of repair until we get to double-strand break repair. So, endonuclease cuts out, and that makes sense because we're in the middle of the genome, not at the the ends of the chromosome. And then the DNA polymerase fills in, and then ligase glues it together. So for BER, could just be that you're missing a base. It's abasic, but it's probably more likely we have a deamination. So C is deaminated to U, and this does not, these don't match anymore. And again, this isn't mismatch repair though, because we know which one is the imposter. If we're the proteins, the repair proteins, U doesn't belong in the DNA. So it is going to be glycosylated, which just means that we're cleaving out that base. And notice that we do that. Now we're just following the exact same steps that we had over here. Now we can just have our endonuclease, and then our polymerase, and then our ligase. Go away. BER now. BER, but a little bit more about the deamination events that we can see. So the deamination is just like a spontaneous ripping off of the ammonia group on our nitrogen base. So if we look at all of our pyrimidines, like it shouldn't, you should memorize a little bit like what deaminates into what, but it should actually make sense, right? We have cut the pie, CUT, cut the pie, cytosine, uracil, and thymine. Who's the only one here with an NH2 group? It's cytosine. If I take this off, what does it turn into? It turns into uracil. Okay. So like, would it make any sense for cytosine to become, guanine, or cytosine to become adenine? No, those are pyrimidines. And it wouldn't make sense for it to become thymine because it doesn't have the methyl group. Who does have the methyl group is 5-methylcytosine. This is 5-methylcytosine. So when this is deaminated, it turns right into that. So that's how you can remember the deamination of the pyrimidines. What about the purines? Well, if you know the salvage in the catabolic pathway of the purines, this will help a bit. Remember that we had ADA, convert adenosine into inosine, and adenine into hypoxanthine, and then guanine gets converted into xanthine. Well, that's all of that's actually going to play out. Those are just deamination events that are coordinated. Here, though, with BER, we're talking about like non-coordinated, just oopsie deamination events. So again, cytosine becomes uracil, adenine becomes hypoxanthine, guanine becomes xanthine, and 5-methylcytosine becomes thymine. Again, remember the fact that you have a methyl there makes, we have that whole long, complicated folate cobalamin pathway, with methionine and SAM and whatnot, to get 5,10-methylene tetrahydrofolate to convert uracil to thymine. So it should really be burned in your head that thymine has the methyl group. So it should be no surprise that the 5-methyl cytosine is going to convert to thymine. Now, in this case, though, it is not easy to tell which base is correct, right? Earlier it was xanthine doesn't belong, hypoxanthine doesn't belong, uracil doesn't belong. So I know that these guys are the original, and these guys you need to get cut out. But in this case, I have a T and a G. Both of these actually belong in my DNA. So I'm not sure which one of them needs to be ripped out. Well, that's where we have mismatch repair come in, and not basic excision repair. Basic excision repair is where we know who the bad guy is. So I always thought of that. All right, so NER now is for bulky adducts, pyrimidine dimers, anything that introduces a large kink, and it's going to be a very similar mechanism. We have an endonuclease. I guess here the difference is you have a helicase to like rip it out, but then a polymerase and a ligase again. One way that I remember the difference between the NER and the basic excision repair is, well, a nucleotide is bigger. Nucleotide is the phosphate, the sugar, and the nitrogen base, and a base is just, well, the nitrogen base. So if a nucleotide is bigger than a base, then it makes sense that an NER rips off more of the lawn than the basic excision. Basic excision was this like tactical strike. We're taking out one little base, one nucleotide. Here we're taking out several. The way that I also reason through this is that this is for a bulky adduct, right? Adducts, here's a benzene adduct, but any aromatic molecules can easily be intercalated. Here's ethidium bromide, but here's this other thing that's way bigger. So because there's a variation in sizes for the things that can get stuck in, it's unlikely that we've evolved such a sophisticated system that it can take out exactly the right length of nucleotides to coordinate with how big the bulky adduct is. So instead, it probably just rips up a whole bunch of the lawn just to be safe and then just refills it in, because it's not that hard and it's better safe than sorry. And it's too difficult to just be incredibly precise with how much we need to take out. This is just one that you might make a connection of ethidium bromide to make gels fluoresce. UV rays can link thymines together. So here we have one thymine and here we have another thymine, and see these two double bonds, kabam. Light can bring them together so that they can now they've formed a bond. And we don't want these two thymines on, adjacent thymines on one single strand to bond together. This is what it looks like. We get this weird kink. Now, this can be repaired with a photolyase, and this is the photoactive reactivation repair. The one that's more relevant here is going to be the NER again, where you just rip off a whole bunch of this shit and then fill it in with polymerase and a ligase. If you have a defect in NER, you can develop xeroderma pigmentosum. You're incredibly sensitive to light because, again, these pyrimidine dimers. So we often say thymine dimers, but could really be in any pyrimidines. I guess you still have the photo reactivation repair, but I suppose that's not enough to compensate for all of the damage that we accumulate on an everyday basis from light. So people get skin cancers all over their bodies. The YouTube algorithm knew that I was looking up like NER last year, and so it recommended me this really sweet and sad video about this girl who's all of these cancers, and just her life is really hard. But then it talks about how she found love and is a good outlook in life. And I don't know, I'm a big sap, but it was both horrible and beautiful at the same time. MMR. So mismatch repair. So this is like during synthesis, right? Because again, we have a mismatch. You and I can say the word mismatch in a conversation, but it's not a mismatch repair if I can tell who doesn't belong. So in this case, I can't tell who doesn't belong. I know they don't belong, but it's hard to tell which of you is telling the truth and which of you is not. Well, there's a lot of mechanisms to identify that the cell uses to mark the new strand. I think in bacteria, they add methyl groups on the new strand, and that's why they know, or maybe they put it on the old strand, or maybe I miswrote the methyls. You don't have to know that, but the idea is we have some way of marking the new or old strand with methyls or acetyl groups or whatnot, and that way, if we have a disagreement, we'll whoever the original was, that's the one that we'll keep. But this is very similar to nucleotide excision repair. And then we have an endonuclease, which is going to cut up a lot of the lawn, a polymerase, which is going to fill it in, and then a ligase to seal the gap. I imagine for this as well that we're going to rip off a lot of the lawn and not have a precision strike, because if you're making mistakes with your polymerase, there might be some weird issues, some skippage, and it might be the case that you're making several mistakes in a row, and that's why it takes out so much of this. I have no idea. That's pure speculation. I mentioned MSH2 and MLH. These are mismatch repair proteins, and if you have defects, you get something called Lynch syndrome. We can get tons of cancers all over your body with a large emphasis on colorectal cancer. Lastly, double-strand break repairs. There is the nice way to fix them, the homologous recombination or homology-directed repair. And that's where, I have a chromosome 17 from mom and a chromosome 17 from dad. And ideally, if I have a defect in one, I could just use the other one and that's that same gene. sometimes we have different alleles, but most of the information is going to be identical. And even if it was a different allele, at least it's close enough that we could swap that information over usually. That's in nature, but we utilize this as scientists to, we'll induce a break, double-strand break, and then we'll flood it with a gene of interest. Either the gene with the mutation corrected, or we might introduce a mutation to study something, or introduce a different gene like a GFP or an insulin gene or whatever. NHEJ is just error prone, and this is just the quicker, faster, easier way to fix something if we can't find a suitable donor that has homology that looks like it would fit into the place there. And this is for generating a knockout gene, or maybe not. It's sometimes scientists will do this for the purpose of generating a knockout gene, and then sometimes just the effect of in a cell is the gene is now non-functional because we put random nucleotides in just to stitch it together. For double-strand break repair, we have two important genes: ATM, ataxia telangiectasia. So you get ataxia, and you get these spider angiomas. So you have these dilated arterioles and capillaries in the face called a telangiectasia. It's said to be blanchable, where, kind of like if you have a sunburn and you press, it'll become white and then it becomes red again. This is because you're pressing down, you're preventing blood from moving through the capillaries, and so it turns, that little section anemic white, and then the blood will refill. If it doesn't blanch, that means you probably have a bleed under the skin. Light causes pyrimidine and thymine dimers, but it causes double-strand breaks as well. So if you have an ATM deficiency, you're going to get double-strand breaks that you're going to have trouble repairing. So particularly sensitive to light is another feature. BRCA, BR, breast, CA, cancer. It also causes ovarian cancer as well. So Ashkenazi Jewish people are particularly susceptible to have a BRCA mutation. Transcription and the regulation of transcription. All right, we're going to talk about gene expression with regard to, what is cis, what are promoters, what are enhancers, how do they interact with transcription factors, what is the transcription mechanism, what about introns and splicing and post-transcriptional modifications, and then regulation, mostly focusing on the lac operon. Why is a muscle cell different than a liver cell? They have the same 3 billion nucleotide code, or 6 billion if you think of the diploid. They have the same 25,000 genes. What's different? It's just the expression of the genes. Which ones are on, which ones are off. What about a normal breast cell versus a breast tumor? Gene expression. What about a normal, like a young breast cell versus an old, 70-year-old versus a 30-year-old's breast? It's just gene expression, right? And if we look at this here, all of this right here represents breast cancer. And this here represents normal wild type breast. These are various genes. And what we're looking at is a heat map demonstrating gene expression. So this color here is going to be lower expression. So if I'm looking at like this particular sector of genes, like this kind of looks like yellowish, like green. So this would be lower expression here. And then if I look at this looks like higher expression. And what I'm seeing is that associated with all of these breast cancer cells, I have higher expression of all of these genes. And with all of these normal cells, I have lower expression of all of these genes. Well, I wonder what these genes are. If we look at this one, this is epidermal cell growth factor. This is an oncogene. CADM1 is an oncogene. CCL4 is an oncogene. And so we can see it makes sense. This is going to be upregulated in the cancer cells and downregulated in the normal cells. And you know what? I haven't really checked these, but you can imagine these are tumor suppressor, in which case it's downregulated there and upregulated right there. So this is just a way that you could quantify and look at. Not that you guys have to really understand anything about heat maps. Cis regulation is anything that's on the same chromosome typically. So talking about enhancers, talking about promoters and whatnot. Trans factors are going to be transcription factors coming from far away, another chromosome perhaps. Typically protein mediated. Often they're going to work together though, right? I have a promoter interacting with an enhancer through lots of these transcription factors in between. Enhancers can be upstream of a promoter over here, or they can be downstream of a promoter. Heck, they can be in the middle of the gene itself in an intron as well. For a promoter, why would we expect to see a lot of AT-rich regions, right? People have heard of like a TATA box as an example. Why all these A's and T's? Well, the promoter, we have this -35 and -10, which is typically where transcription factors are going to bind like their two arms. And the first thing we have to do is unwind the DNA to allow everything and allow transcription to occur. And it's easier to unwind and sort of melt or to unanneal two strands of DNA if they're not bound as tightly. Well, remember that CG has 3 and AT has 2 hydrogen bonds. So it makes sense that AT is disproportionately concentrated in the promoter. So we are going to have our promoter and maybe our TATA box, and we're going to have a TATA box binding protein and then a couple of transcription factors and then more transcription factors, and eventually they are going to recruit the polymerase. Everything is unwound, and the polymerase starts moving to the right, to the right, to the right until it hits this +1 site, and that's where it's going to start incorporating nucleotides. Nucleotides that are going to be identical to the coding strand, but which are perfect, but are literally measured up against the template strand. So they match the coding, but they're measured up against the template. And because it's the antithesis of the template and the coding is the antithesis of the template, then that means the mRNA is actually the same as the coding. Keep in mind, this is upstream and this is considered downstream typically. So what I was just saying, we can see a lot clearer here. Remember, this is 3' and this is 5', which means this would be 5' and this would be 3'. We are transcribing in the 5' to 3' direction. So here's my coding strand. We have TAC, and the mRNA we see is going to be UAC. It's identical except for that T's become U's. But we get this by, it's not ever actually even seeing the coding strand. It's literally copying the template strand, also known as the anti-strand strand, also known as the non-coding strand. So that is where we're literally laying down these new nucleotides to generate our mRNA. Transcription occurs first, and then after that, translation. If we look at something like this, we see this is our mRNA. We better be able to infer which one of these is the coding strand. Boom, it's the one that looks exactly the same as the mRNA, except for T's become U's. We have to splice out introns. So I think of this as like the opposite of the Snyder Cut, right? He had this, we had a short movie that was like 2 hours, and he said, I'm going to put in all of this extra deleted scenes and footage to make it really long. It's the exact opposite. Here we're cutting out all of the footage that no one really wants to see, and we're making it a nice, tight, condensed, final product with no extra garbage. So it's exons only. So the exons are ultimately expressed. We're going to come back to these UTRs as well in a hot second. So keep that in mind. Bacteria do not have introns. So if we want to splice an insulin gene from humans into bacteria, you have to cut out all of the introns first. Typically, you do this by transcribing it into mRNA, reverse transcribing it into cDNA, and then you can put that into the bacteria because they just don't know how to. They'll try to express the introns as if, they'll try to make amino acids out of them, which will be nonsensical. So this is for introns to understand the splicing. A nucleo, is like an SN2 reaction, comes here, and then that leaves, and we get this, right? This picture actually stinks. I think this is in first aid. Technically, that should go over there, and there's a couple other things about this that are misleading. So let's just go ahead and look at this. So what we're seeing right here is this picture right here, this third one. So here I have my spliceosome. Here I have exon 1. Here I have exon 2. And we have something called a branch point. We're going to have an adenine. Nucleophilic attack. This is going to attack and cause this. So it's going to attack sort of like right here. And this is going to then be our leaving group and go right there. So again, we have our, this on its own. And again, the adenine is just attached to the phosphate. And so now this OH is going to go ahead and attack there. And then that's going to leave. And boom, we now get a lariat just like that. This is the exact same as group 2 self-splicing introns, except we do not have a spliceosome. It's the exact same thing over here. This attacks there. We get a leaving group. The thing that just left now is attacking exon 2. It's connecting to exon 2, but it's causing this to leave in the process. And again, we get a cyclical lariat. For group 1 self-splicing, we have an exogenous guanine. So an exogenous guanine floats over. It's not actually in the nucleic acid structure of the intron. Again, it's exogenous. It's like a monomer. It just happens to be close by. So it goes over. It attacks. It just literally attaches to that. Of course, that is was a leaving group, did leave, and then the same final step. This attacks there and causes that to go over. But because again it was exogenous, we do not have the cyclical lariat form. We just have this kind of linear structure right there. So there's a lot of similarities between them. There's much more similarities than there are differences. So I would just try to keep that in mind as you review the splicing. Alternative splicing is where it's one man's intron is another man's exon is one way you could think of it. So in this case, we have 5 possible exons. Maybe one version of the protein gets all 5 exons, and that could be a protein in like a muscle cell. And you know what? Maybe in like a bone cell, for whatever reason, we don't want exon 3. And so, we have just exons 1, 2, 4, 5. And, now we don't want exon 4. We have 1, 2, 3, 5. So that would be, it'd be like we're taking, all of the footage for the Zack Snyder thing, and we're making like 10 different versions of Justice League movie based off of like which deleted scenes we wanted to keep in or not keep in. So this way you could have one gene that could potentially generate hundreds or even thousands of different versions of a protein based off of the different combinations of exons that you allow in or potentially exclude. So in addition to splicing, we're also going to add this 5' guanine cap and this poly A tail. The purpose of this is to export it from the nucleus. Getting out of the nucleus is like getting into a club, and you got to get past the bouncer, and the bouncer will only let you into the club. I guess the club now is the cytosol. We want to get out of the nucleus. You need to have the 5' cap and the poly A tail to be let out. RNA is incredibly unstable, right? Because we have this 2' OH group because it's single-stranded. This gives it extra stability because we also have exonucleases. Exonucleases, keep in mind, as an endonuclease will come in here and induce a cut. Exonucleases will try to eat up at the 5' or eat up at the 3' end. But if you have a poly A tail and you have a 5' cap, it protects you. It also helps us latch on to ribosomes for translation. All right, this is everything together. So we have our open reading frame starting with AUG. The end of the reading frame is going to be UGA or AUG or UAA, or enhancer and a promoter. So we have the +1 site, which is right here, and we're going to transcribe everything. And note that when we cut out the introns, we still get these. We have these little black regions. So this is going to be a UTR. UTR is an untranslated region. And you might say an intron is an untranslated region, but yeah, an intron that was cut out during, right after transcription. So an untranslated region is technically something that survives the transcriptional processing, makes it out as into the mRNA, but is nevertheless not translated by the ribosome. So that is a 5' UTR and a 3' UTR. And actually, sorry, the UTRs are right here. I actually misread that. This is just showing you the start and the stop. There we go. Cool. Bacteria transcriptional regulation. So let's say that I'm not eating milk because it's giving me bad tummy issues. So every 3 days I am ingesting milk, but that's not that often. So the bacteria in my stomach, over here, it doesn't make sense for them to, what color should I make milk? Milk will be blue, I guess. Blue milk, like Star Wars. So if I'm not making milk, if I'm not consuming milk, the bacteria in my stomach don't want to waste time and energy generating the enzymes necessary to digest milk. It's only when I ingest milk and it comes down to them that they then decide to activate the production of the machinery necessary to process milk. And so let's take a look and see exactly how that happens. Here, so this is going to be far away. This is far away. We have an LAC inhibitor gene far away on the genome. And then from here to here, we just have our whole LAC operon, which includes things like our promoter. It includes things like our operator, which can be switched on and off by the repressor. And then we have a couple of genes which are bundled together This is a feature that bacteria have that humans don't have. We're monocistronic, one gene, one protein. They're polycistronic. Several genes can be transcribed sort of at the same time as part of like one unit. Typically, they're going to have they're going to be functionally related. So far, far away in a galaxy long, long ago, we have an inhibitor made right here. And if we just think like third graders with the shape of blocks, this shape could perfectly fit right there. So this is the natural state of affairs. An inhibitor is made that perfectly fits into the operator. It binds, and it's now a giant roadblock. We now have this, giant roadblock thing. And you know what? My polymerase here cannot get past this roadblock, this big clunky repressor, which is plugged into our operator. And so what happens? I do not get transcription of LAC-Z, beta-galactosidase needed to digest milk. LAC-Y, which permease lets the lactose in. And the transacetylase, no one really, I think, fully gets what that does, so we'll ignore that one. But this makes sense, right? We just think economically, would it make sense for the bacteria to try and make the enzymes to digest milk if I don't have any milk? No. So this is, and I'm not normally, right? We said I only have milk every couple of days. So this should be the normal state of affairs. All of this stuff should naturally up. This should make sense. Now, I have like mocha frappuccino and there's lots of dairy in there. What's going to happen? I'm going to have milk. It's going to come in. It's going to go through my GI. It's going to go to the bacteria. Let's just think milk is lactose and lactose is allolactose. So this little milk particle right here is going to come in, and the repressor has been made so that it perfectly fits one of these little milk molecules. And what happens, just again, we're third graders with blocks, what happens when a milk molecule binds to this repressor? It changes the shape. So now does this shape, I wish I can I actually undo this? Whoa, we can't undo it. Great, so we're third graders again. Does this shape right here, does this fit into there? And the answer is no. No, it does not. What we've done is we now inactivated the repressor. And if I cannot, if I can't repress this, then what's going to happen? What's going to happen is we're now going to have our polymerase is open for business. There's no roadblock. It's going to transcribe this, and this. We're going to get all of these three enzymes. What are they going to do? They're going to help us digest milk, which makes sense because I have milk. You can only digest milk when milk is there, repressing the repressor, allowing transcription of the things necessary to digest milk. This will go, it will chew up all of the milk. It will convert the repressor back to its normal state, which makes sense because we just consumed all of the milk. We want this to go back in. This is a smart system if you think about it. So that's bacteria being like lazy or smart or economical. But we could also say that they're picky. And you could also actually, so weave this into the economics like metaphor. The idea is that they will only digest milk if there's milk there. But at the same time, they would prefer glucose. So if there is milk and lactose, they will just go for the milk, and they will probably not make much of the enzymes to digest lactose. Now, how does this manifest? Glucose is going to indirectly correlate with something called CAMP. So if you have low glucose, you have high CAMP. If you have high glucose, you have low CAMP. And cyclic AMP is needed to activate the CAP transcription factor. So if we look at this, we have a high lactose. High lactose simply means that the lactose bound to the repressor inhibited it. It doesn't bind to the operator, so the operator is free. Transcription can theoretically occur. It doesn't mean it will occur at a high rate. High glucose is there too, though. Let's just think about the economics. Do we really even want to waste time making all of this stuff that I just lactose if there's glucose there? No. And the way that this physically works is that high glucose correlates with low cyclic AMP, which means the CAP will stay inactive. You do not have this transcription factor. You will not get much transcription. On the other hand, if we look at this, high lactose, the operator is free. Low glucose means high CAMP, which means CAP is active. We can have transcription, and it will happen quite quickly because of the CAP protein. So 4 possibilities here. If we have, just think about the economics, and then we can think about what's actually going on with the molecules. Low glucose means high CAP means CAP is active. Lactose is available means the repressor is bound, and so it's not doing its thing, which means the operator is free, which means transcription can occur. This is the scenario where you have a lot of the lac genes expressed to digest the milk because there's milk and no glucose. High glucose and lactose. This is a double no. We can just go for the glucose, and it wouldn't even make sense to make the lactase enzymes because there's no lactose. So, nope. Low glucose and lactose is unavailable. Well, yeah, there's no glucose, so it would be nice to digest milk if we had milk, but we don't even have milk, so the repressor is going to be bound really tightly. We're not going to waste ATP and time transcribing this particular operon, so it's not expressed. High glucose and high lactose. This is where the bacteria can afford to be picky, and they go, yeah, I know that theoretically we could transcribe all the lactase stuff because the operator is open, but you know what? We just rather go ahead and digest glucose. So the low CAMP levels means that CAP is inactive, so you'll have a very low level of expression there. Into a little summary, just go ahead and take a mental snapshot or just stare at it forever. All right, so RNA is very unstable. We said that single-stranded 2' OH group. So we have a couple different kinds. rRNA is actually 85% of it. I would definitely know this. So the most ubiquitous type of RNA is going to be the ribosomal RNA. It's made in the nucleolus. tRNA is 10%, and then mRNA, which really gets all of the accolades, is the least common one. Everything is 5' to 3' except for exonuclease. Type 1 for rRNA, type 2 for mRNA. So this is probably the most important one to know, even though it's only 5%, just because, I don't know, it's important. Amanitin it's from a fungus. It's an inhibitor of the type 2 RNA polymerase. And then type 3 is for TNA, but it actually, it does make, not all of the rRNA is made by type 1. A little bit, the 5S rRNA that goes into ribosomes is by type 3. Rifampin, something we use for tuberculosis, is an antibiotic. It's going to inhibit transcription. And then actinomycin can affect both eukaryotic and prokaryotic transcription. So this could be used, if against bacteria as an antibiotic. But if we want to stop eukaryotic transcription, we might use that if there's a lot of transcription, a lot of replication in a cancer. All right, last section, we're going to talk about RNA interference, which is known as knockdown, and CRISPR. Now, CRISPR can be knockout if it's teamed up with NHEJ, but if you do it with homologous recombination, well, this is where the whole genetic engineering thing comes into place. We can, swap genes effectively. So RNA interference is all about inhibiting translation. So exogenously, we could introduce double-strand RNA, which makes siRNA, or sometimes cells might produce their own little hairpin RNAs known as microRNAs. Both of these can be cleaved by dicer and then team up with a complex of proteins known as RISC and Argo. Argo is an argonaut, and you can actually see a better picture over here. So the argonaut RISC complex teamed up with this little guide RNA, this siRNA over here, and the microRNA with the little hairpin over there. And if you know anything about Greek mythology, like Jason and the Argonauts, they went on all of these insane adventures all over Greece, all over the world. And I think that's what the argonaut protein is doing. It's taking this siRNA, this miRNA all over the cell, and it's introducing it to various pieces of mRNA that it meets, seeing if they're a match. And I think of it as Barney introducing Ted to random girls at the bar. The Argo RISC complex introducing the miRNA and siRNA to random mRNA in the cell. If there is a match, if they fit together, it then goes ahead and it inhibits translation of that mRNA. Now, in the siRNA, it is important that there is a complete match between the mRNA and the siRNA that we have right there. And if this is the case, then we cleave the mRNA. So again, we're all about inhibiting translation. Well, if you cleave the mRNA, you're absolutely going to inhibit translation. In this case, notice that it's not a complete match. It's a partial match. And instead of degrading the mRNA, it's usually going to repress translation. So miRNA is a partial match, and it typically represses translation of the ribosome. And the siRNA is a complete match, and it typically results in cleavage of the mRNA. CRISPR. So what's all the hullabaloo at? If a virus infects a bacteria and injects the viral information, and this information, let's say the bacteria lives, this information can be stored in the genome between these little palindromic repeats that we see over here. And the idea, why store, you got attacked by a virus, you lived, why store that genetic information into the genome? Because the virus kind of uses this genetic information, it sort of is like a mugshot. It's like an adaptive immunological system, an adaptive immune system that remembers previous encounters. So that if it now gets attacked by the same virus again, it can go ahead and transcribe this RNA called CRISPR RNA. It teams up with a pair of proteins, and they can find the invading viral genetic information, and they can cleave it. And if they cleave it, then it can't really do its job. So each of these colored things here is the remnant of a previous battle, right? We might have, flu pox and chicken pox and HIV and I'm of course kidding, but any type of virus can be stored in here. Those are the spacers, and then the palindromic repeats. This is actually where CRISPR gets its name. Clustered. This is all clustered in one part of the genome. Regularly interspaced. So that's a regular interspace, and that's a regular interspace, and that's a regular interspace. Short, relatively short, and then a palindromic repeat. It's ugly is all sin right here, but this little black thing here, you can see that these are in fact palindromic repeats stored in these black areas. Necessary the hairpin so that the Cas9 protein can get a handle on them. So this is getting pretty friggin' ugly. So let's just go ahead and look at this. So again, we are a virus is attacking. We're storing, we live to see another day. We store that genetic information in between these palindromic repeats. So where the viral information is known as the spacer. This can be then transcribed to make a guide RNA, which technically is CRISPR RNA, but we'll get to that in a second. This can team up with Cas9. So the Cas9 genes are elsewhere in the genome. So we're making the Cas9 protein elsewhere, but that's teaming up. And honestly, what this is very much like is that RISC-Argonaute complex. Cas9 is like the RISC-Argonaute complex, where it is introducing the guide RNA, the CRISPR RNA, to the potential, viral, DNA match. Seeing if it is a match, then we're going to go ahead and kind of like we saw with the siRNA, we're going to degrade you, we're going to cleave you. Here's another picture just so that you can really wrap your head around it and really see. So again, here is our Cas9, and here is going to be the spacer, which is the previous viral mugshot. And here's a new virus injecting this information, and we're going to see if there's a match. And if there is a match, have you met Ted? Yes, you have met Ted. You guys are a match. So I'm going to go ahead and cleave you then. There's a lot of different types of the CRISPR-Cas9 system. The type 2 is probably the most common one, and this is where, in actuality, the Cas9 is teaming up with the CRISPR RNA and something called tracer RNA, which sort of acts as like a handle that it can hold on and get a better grip on everything. And so you can just get a better picture of this here about how the CRISPR RNA teams up with the tracer RNA. Right here on the CRISPR RNA is the previous viral sequence, the spacer, that was stored. And again, we're trying to match this with the newly invading viral sequence, which is really technically all of this, but this is probably the only part that would match. And so that the Cas9 can go ahead and induce these cuts and kill the invading virus. So the CRISPR and tracer RNA used to be two separate things, but scientists realized, it might actually just be easier to make this one giant RNA that we can just tinker with the sequence that's supposed to complement the genetic information that we want to seek out. And in this way, we can do genetic engineering experiments a lot more easily. So typically now we could just call this single guide RNA. So kind of the whole thing, we're going to design our guide RNA of the CRISPR sequence. and maybe we want to edit the insulin gene to put like a GFP in. Maybe we want to turn it off in one case. So let's say this is the insulin gene here, and we want to turn this off. So we've designed a guide RNA that has something that matches one of the insulin exons. So there we go. This is we edit the guide RNA, but this is really the part that we're editing the most, this little box here. And so we've created this. Cas9 will now heat seek like a missile. Have you met Ted? Have you met Ted? It goes all over the genome. Have you met Ted? Until it finds the insulin gene. And aha, we have met Ted. And maybe I'm trying to destroy the insulin gene. So this is all we do. We just have the CRISPR and the Cas9 that match with the insulin gene. It makes a cut. And if you make a cut, probably what's going to happen 99 times out of 100 is NHEJ. And now we've knocked out the insulin gene. Great. That's one experiment. Maybe instead now I want to put in like a GFP because I want to, or maybe I want to put in some mutation because I just want to see how does a different allele of the insulin gene, what would this do in this organism? So what we have to do now is we have to flood the cell. As it makes the cut, we need to make sure that there are tons of copies that look very similar to the cut region, right? There's a lot of homology, but of course it has to have that little one mutation that we are interested in to seeing what the effects would be. So we have to flood it with all of these similar insulin-esque gene copies for the region that we're cutting of that exon. And what will probably happen now is homologous recombination, where we're, rather than just putting in random shit, which is NHEJ, we're putting in this extra genetic information that we have supplemented. In terms of gene expression, if you do a knockout, you completely abrogate gene expression. You should get none. If you do knockdown or like siRNA or even miRNA, you should get less, though I suppose you could titrate this and do more or less depending on how much RNA interference you do. And of course, this is all in reference to our control. All right, that's everything. I hope that was helpful. All the best to everybody.